The overall goal of this procedure is to differentiate mouse embryonic stem cells to a neural lineage in a serum-free monolayer cell culture. This is accomplished by first coating the culture vessel with gelatin. In the second step, the embryonic stem cells are dissociated into a single-cell suspension in differentiation medium and diluted to the appropriate plating densities. In the final step, the cells are seeded onto the gelatin-coated plate. Ultimately, immunofluorescence microscopy is used to confirm the success of the differentiation by visualization of the neural markers of interest.
Generally, individuals new to this method will struggle as plating at the appropriate density is critical to achieving a good differentiation, and there are several factors that affect the plating density both directly and indirectly.
Before plating the cells, warm an aliquot of 1% gelatin in a 37°C water bath until dissolved. Then mix the gelatin with 500 ml of warm PBS and cover the bottom of the cell culture container with just enough of the solution to coat the surface. While the gelatin is coating, rinse the subconfluent culture of mouse embryonic stem cells 2 times in PBS. Then add 1 ml of dissociation reagent to the culture. After 2 to 5 minutes, tap the vessel to dislodge the monolayer into a single-cell suspension and collect the culture in a total of 10 ml of medium and cell dissociation reagent. Transfer the cells into a 15 ml conical tube and spin them.